Hello and welcome to the Farming Today podcast with me, Charlotte Smith. In this episode, how DIY is helping keep an ear on bats and a new growing technique which could help save the ash.
And then you have to try and get the scalpel bleeds. It's just about trying to peel it apart.
I've never worked in a lab.
Right, there we go. You
there
can see it.
is. Oh, wow. I feel like a midwife.
More from Anna later. First, campaigners are calling for a full-scale clean-up of beaches on the Isle of Sheppey in Kent, following the discovery of a large quantity of illegally dumped clinical waste. We've been highlighting problems with illegal waste dumps and fly tipping around the UK. In this latest dump, hundreds of vials labelled virus transport medium are believed to be among thousands of tonnes of waste dumped over the cliffs at the north of the island. The local MP says he's deeply concerned and has called for the Environment Agency to start an urgent investigation. Yvette Austin has this report.
Waves lapping the shore on the Isle of Sheppey's northern coastline, a 750-acre site of special scientific interest, an important wildlife habitat famous for its fossils. But on closer inspection, all is not well. Small, bright-coloured pieces of plastic are scattered almost everywhere you look, and even more concerning, clinical waste too. Plastic test tube-like vials with red lids. Fossil hunter Daniel Hogburn has been finding them spread across the beaches. They're labelled virus transport medium and are potentially a biohazard.
So this is one of the bad piles, obviously. You can see them accumulating at the high tide line. There is, hundreds of them.
You can see.
So obviously tops, vials. One there which is, something very nasty. Another one there.
It's believed the vials were among the many lorry loads of waste that were illegally dumped over the edge of the cliffs here at Warden Point over a number of years. Waste was also dumped a short way along the coast at Eastchurch Gap until the Environment Agency shut both sites in 2023.
It's soul destroying. When the sort of dumping was live in both locations, all you could hear was lorries reversing up, tipping, diggers clanking away, mixing the soils, the rubbish.
It's possible the vials could contain patients' virus swab samples, or they could be unused. They have liquid inside in varying colours, from clear to red. The MP for Sittingbourne and Sheppey, Kevin McKenna, who has a degree in microbiology, says an urgent investigation is needed.
This is horrific. We need to find out what's actually in these, so I'd ask the Environment Agency to go there really quickly and investigate those samples. And then, previously, the Environment Agency said it's too expensive to clear the waste from the area. It's also very dangerous because it's by eroding cliffs. It's actually very hard to access. But honestly, we now got clinical waste that's being washed up as well. I've also heard reports of asbestos in the water. This needs to be looked at again, and we need to see if we can clean it up properly and make it safe.
A view echoed by Daniel Hogburn.
It needs to be cleared up. It's a biological hazard. So what's to stop a child picking up one of these, opening it up, and dare I say, drink it? It's just disgusting.
Any clean-up would be extremely costly. But 32,000 people have now signed a petition for the removal of the waste, and the authorities are under increasing pressure to act.
Yvette Austin reporting there. An Environment Agency spokesperson said, "We are determined to turn the tide on waste crime, getting even more shameless waste criminals to face justice for their blatant disregard for local communities and the environment. With more drone flights to track crimes from the sky, more officers on the ground and stronger partnerships with police and councils, to those committing waste crimes, we say we will stop you." Now, all this week here on Farming Today, we are listening to the countryside. You might expect to hear the lowing of cattle or the baaing of sheep. We are taking a bit of a deeper dive, literally in some respects. Today, we're talking about bats. Scientists in the east of England say a DIY approach is helping to expand research into bat populations. The University of Suffolk and a local group have been monitoring bats using bioacoustics to track their ultrasonic calls as they visit people's gardens. But they didn't have enough microphones to go round. Well, now they're working with people to build their own at a fraction of the cost, which means more kits and more data. BBC Look East's Zoe Dennis reports.
How do you track an animal that doesn't want to be found? For scientists at the University of Suffolk, the answers might be in the hands of the public. Building low-cost microphones that can pick up the ultrasonic calls of bats. Dr. Bowler is one of the project leads and says it's been making wildlife conservation more accessible.
The microphones we've been using for the last 5 years, they stay in people's gardens for 2 weeks at a time, and then we get lots of files back to analyse in the lab. Unfortunately, we haven't had enough microphones to get around all the people that want to have them in their gardens. So these new microphones that we get people to build themselves are cheaper, and we can scale it up. We can get people building their own microphones, keeping the microphones, and we can reach more people.
So the recordings from the microphones, their results come back here to be analysed. What exactly are you looking for in that data?
Well, these recordings have been sorted by AI. So they've been sorted by species, but it's really important that we manually verify each one. And by measuring the dominant frequency in the calls and looking at the shape of the calls, we can identify the species. So this is at 45,000 hertz, and it's the shape of hockey sticks. So we know that that's a common pipistrelle.
Cameron and Grace study wildlife conservation at the university and volunteer on the project.
So I volunteered for this project initially because, my love for bats and getting a stepping stone into ecology after uni, and hopefully to see where it goes from there.
My favourite part of volunteering has been going to the workshops and having the opportunity to interact with the public and teach them about it.
And to get that data, the team is turning to the community in the village of Melton, helping residents build their very own kits.
So I've got a broader interest in the wildlife and things like that, not necessarily bats specifically, but that sort of brought me in.
I was really fascinated that there are bats that they thought were rare, and actually they're not rare around here at all. It's just they're widely known about till the detectors were used.
With bat populations in decline, researchers say rural community involvement is vital to turn simple recordings into targeted conservation.
Zoe Dennis reporting, and we would love to hear your sounds of the countryside. Just record whatever it is that you love to hear and send it to us on an email. We are farming.today@bbc.co.uk. Please keep it short, or the BBC firewall will just ping it back to you. Tim Collins has already sent us this. He describes it as the Fife version of the Dawn Chorus. And Jim Sadler sent us this. He says, this blackbird visits me every spring. It sits on my beech hedge, singing its heart out all day. So please do go and have a listen around the countryside and send us what you hear. Our email again, farming.today@bbc.co.uk. A new propagation technique could help restore the ash tree to our landscapes. 14 years ago, the first trees with ash dieback were found in Norfolk by a plant scientist volunteering in a community woodland. Since then, hundreds of thousands of trees have died, but some have proved to be resistant to the disease. Scientists across the UK have been working to propagate more trees from them, but currently that takes between 2 and 5 years. Well, now researchers at the John Innes Centre have developed a speedier system, which they hope everyone could use, even at home, with the help of household bleach. Anna Hill has been to try out the new technique. She met up with Dr. Anne Edwards, the person who first identified the disease.
It was absolutely devastating. We'd had reports from Denmark, who had already had the disease for about 10 or more years, and they were saying we've lost over 90% of our ash trees. It was just a very depressing time, actually, very, very welling up at the thought of it now. Yeah, it was a very depressing time and a worrying time, yeah. It was terrible.
Looking then over the next 14 years, where we are now, are you pleased with the amount of research that's been done?
Delighted, yeah, there has. There's been a massive effort looking at the genetics of ash and then also finding resilient trees and propagating those.
And now we're going to look at the latest step in this process, which is actually asking ordinary people like me to help and maybe even start propagating resistant ash trees.
That's absolutely fantastic. I mean, people do love trees. You only have to think back to the horror of the felling of the Sycamore Gap. It's a connection with history, but it's also looking to the future. And you know, they're a powerful weapon now going headlong into climate change. They are our friends and we have to take care of them.
So we're just going down to Elizabeth's lab.
I'm Dr. Elizabeth Orton and I work in the crop genetics department at the John Innes Centre. This disease has really been very devastating in the wild, and we're seeing huge losses, thousands, and we'll get into the millions of ash trees are being killed. It's stopping recovery, natural regeneration of ash in woodlands as well. But we're now seeing that some trees are much, much healthier in the wild. In areas where there's high disease pressure, so that what means we're seeing lots and lots of dead ash, we can really now identify those resistant trees.
The latest thing is that you have found a way to speed up the process of regenerating ash trees.
Here we go. So basically once the seeds have been soaked in the chemicals that sterilise and soften the seed, what you can see on this plate of the agar jelly in a Petri dish is the seeds being left on there overnight, but I cut just the edges off the seed, and that just helps again with the whole kind of softening process. So it should make it really easy for us today to get one of these embryos out. There we go.
Wow.
There we go.
You've peeled it open, so the long, thin seed casing has now been broken open, and inside is a bright white little seed, and that is the embryo?
Yep, so this is the little embryo of ash. Yeah, as you said, so at the top here we've got the two what are called cotyledon leaves, so they're the very first leaves, and a little stem. It doesn't have a root yet, so that's why we put it onto the nutrient agar, and after about a week the little leaves have greened up because they're starting to produce chlorophyll, and we start to see a little root coming out the bottom, and I just think that's amazing.
Am I allowed to try to do one myself?
Of course you can, yes.
You'll have to hold the microphone.
Oh dear.
We're swapping jobs. So I've got the seeds.
And then you have to try and get the scalpel blades. It's just about trying to peel it apart.
I've never worked in a lab.
Right, there we go. You
there
can see
it
it.
is. Oh, wow. I feel like a midwife.
Yeah, sometimes you can just slide them out very, very gently. There we go.
I've done it. That is so satisfying, isn't it? I can see that this would be possible at home. People have to wait till you've got your instructions absolutely sorted out.
I've been trying to get the chemical process down at the moment. One of the problems is that, they can often actually be quite strong household chemicals, and it's quite hard to get the right level. So I've probably overdone it on a couple of occasions and bleached them to death, quite literally. So this is what I'm trying to get the balance right. And also we need to consider safety in the home as well. So yeah, we do have plans to put out a blog and maybe even a little video as well that we can show people. I think it's nice for, potentially younger people to see this happening and really get them engaged. So if we can speed up this normal natural selection process, I think that would be fantastic.
Dr. Elizabeth Orton from the John Innes Centre with Anna there. And that's it from us. I'm Charlotte Smith. The producer is Beatrice Fenton, and Farming Today is a BBC Audio Bristol production.